It's now my distinct honor to introduce Dr. George Church, Professor of Genetics at Harvard Medical School and Professor of Health Sciences and Technology at Harvard University and at the Massachusetts Institute of Technology, MIT. Dr. Church is Director of the U.S. Department of Energy Center on Bioenergy at Harvard and MIT and Director of the National Institutes of Health Center of Excellence in Genomic Science at Harvard. He is also the director of personalgenomics.org, which provides the world's only open access information on the human genome, environmental and trait data, known as GET, G-E-T. Dr. Church is widely recognized for his innovative contributions to genomic science and his many pioneering contributions to chemistry and to biomedicine. In 1984, he developed the first direct genomic sequencing method, which resulted in the first commercial genome sequence, the human pathogen H. pylori, dramatically reducing the cost and time of sequencing genes. This led to his participation in the founding of the Human Genome Project in 1984 and the Personal Genome Project in 2005. His career ever since has been a string of revolutionary firsts, not only in genomic sequencing, but as one of the pioneers of CRISPR technologies, synthetic biology, DNA data storage, privacy, safety, and security policies, nanotechnology, and innovations in aging, brain research, and even dark matter and space genetics. His work includes research on organ transplants, endangered and even extinct species. He is rumored to be working on bringing back the woolly mammoth and on large ecosystem gene drives, the subject of his talk tonight. Dr. Church invented the broadly applied concepts of molecular multiplexing and tags, homologous recombination methods, and array DNA synthesizers. He has co-founded more than 14 biotech companies, authored over 400 papers, and holds 60 patents. He is a member of the National Academy of Sciences and the National Academy of Engineering. He has received dozens of awards, including the American Society for Microbiology Promega Biotechnology Award, Research Award, and the Heptannual Bauer Award and Prize for Achievement in Science from the Franklin Institute. In 2012, Dr. Church authored the book Regenesis: How Synthetic Biology Will Reinvent Nature and Ourselves. A recent National Geographic story about Dr. Church states that he has made a career of defying the impossible. Time magazine recognized him earlier this month and named him one of the 100 most influential people of the year. Be sure to read Stephen Colbert's description of him in Time if you get a chance. It was quite interesting. With that, Dr. Church, please come forward.
Thank you very much, both of you, for that introduction. So this is my conflict of interest slide, which is also my thank you for all the effort that it takes to go from a crazy idea to reduction to practice and getting it out into society where it can have some impact. And I want to talk about engineering human genomes and environments, which requires we not only engineer, but we have to be able to test and read them. And this is a very interesting story. I know plots may seem quite dry, but this is factors of 10 on the y-axis. This is not only exponential, this is super exponential. This is going not only faster than Moore's law, but it keeps getting faster. And so something remarkable happened around 2003 where we started getting, we've now improved the cost and quality of reading and writing DNA by 3 million-fold for reading DNA and 1 billion-fold for writing short pieces of oligonucleotides. Now that's not the same as getting it fully functioning in a medical or agricultural or environmental setting, which is what we'll talk about. What happened in around 2003 is we learned the lessons of Silicon Valley, of microscaling and multiplexing, and we just applied them to biology. Maybe a little more than that. Quick audience participation. Is human genomics useful today? I'll argue probably not. So raise your hands if you have your human genome in your possession. Well, it's actually, that's some surprise. There are quite a few people. Okay. This is what the rest of you are missing out on. It is helpful for preventative medicine, essentially for avoiding, and in some communities that practice genetic counseling, almost eliminating very serious inherited diseases like Tay-Sachs. It's not fully appreciated that about 5% of births, even in very
wealthy nations, 5% are children that are severely genetically damaged, and their illness could either result in very early death or lingering and up to a $20 million lifetime costs of that disease. There are later steps. This is, in vitro fertilization, prenatal diagnostics very often results in a dietary change during newborn screening. And then sometimes children have something that's just baffling to medicine, and that's called a medical odyssey or genetic odyssey, where the parents go from place to place. They blame themselves until a cause is found. Even if there's not a cure, knowing the cause is very reassuring to the parents, and it sometimes results in research, and sometimes, in the case of the Beery twins here, these beautiful teenagers were misdiagnosed with cerebral palsy. It turned out all they needed was an over-the-counter nutritional supplement for dopamine and serotonin pathways. And then finally, there are adult onset diseases, and Angelina Jolie was sort of very helpful in prominently being transparent and disclosing her dealing with cancer. This is just to make sure that nobody misunderstands. This was not she detected a lump or that she detected mammogram positive. This was entirely preventative. She had no cancer. She just had a predisposition. So she reduced her risk from almost 90% to about 5% by preventative surgery. So what about prevention? Is that the future of the biomedicine? Well, if it's not reflected in the budgets all the time, this is a pie chart showing one of the national components, and here 1% of this NCI budget is for prevention. Nevertheless, a huge fraction of cancer actual on the ground that works is preventative, avoiding UV, smoke, certain pathogens, and so forth. Now, speaking of pathogens, I'm recovering for 11 days of some respiratory illness, and when you see someone cough, you wonder, gee, is that nothing? Is that non-infectious, like a allergy or neuroreflex, or is that, some deadly disease heading my way? And I think when we start talking about environmental components, and I will talk about environmental engineering, it'd be nice to have a real-time wearable device that tells you exactly what's going on in your air, your water, your food, your environment. And there is something that's close to that, which is this nanopore technology. There are two nanopore technologies. The portable one here, the handheld one, is Oxford Nanopore, and there's another one in development by Genia Roche. And this is actually not quite real time, but it's pretty close, 25 minutes from the time you put in your sample to getting the first DNA sequence. Now, that's reading your environmental components, and microbiome is a huge fraction of your environment, but you can also engineer your microbial environment. And here's an example of 5 companies I happen to be involved in that deal with everything from detoxifying, if you have some toxins in your body, either due to inborn errors of metabolism and so forth. There's skin health. There's actually some of the brain metabolites are made by the microbiome. Your microbiome is in your gut, is in equilibrium with your body. That's why many of these things happen. And finally, if you take antibiotics, you can end up with way too much Clostridium difficile, which is a serious dysbiosis, which you can fight by fecal transplants, which are not very well controlled. They're typically, somebody who has some poop that you can use. And Ceres is trying to make this something that FDA can get behind, a reproducible. Now, there's, in addition to the sequencing method I described, that portable sequencing, there's another revolutionary kind of sequencing, excuse me, which is our ability to read in situ. And this is an example of a bulkier instrument. It's basically a modified microscope, a custom, that allows you to read every part of a cell. DNA, RNA, and protein. And you can see each of these dots is changing color, not position. Its position is fixed. Then the computer displays it in a way that shows that each of those dots now can be read out as a barcode. If you take all the time axis from the previous steps, you can see that. Sorry. So you can do this. Part of the BRAIN initiative was inspired by this new technology where we can get that kind of fluorescent multicolor readout. Over very large distances, you can track these very large projection and connections, or connectome, throughout the brain, and we're trying to do this to map out the connections, the expression of what molecules are made in the different parts of the brain that makes one cell different from another, and the activity that is associated with the program as it executes in a living, behaving brain. This is work from Tony Sater's group, with whom we're collaborating on this BRAIN Initiative project. Now, you've probably all heard of CRISPR. Somebody raise their hand if you haven't heard of CRISPR. There. It's fewer than the number of people who've been sequenced. But I think my team and my ex-students have been involved in most of the transformation of it from a discovery, basic science discovery, to a technology. But I'm going to downplay it a little bit. I'm going to say that's not the revolution. There is a revolution, no doubt about it. I showed you those exponentials. The revolution is about omics, genomics, transcriptomics, proteomics, microbiomics. And it's not just about, it's about reading, writing, and testing, all integrated. They have to be all integrated in order to have an impact on society. We've launched a new project that goes far beyond editing to writing. You can think of editing as you change one letter in a book, in best cases. But what about writing the book from scratch? What can we do there? Or what can we do? Can we do editing by being able to write anything we want? And so one of the things we're trying to do is make human cells that are already used, mammalian cells already used for manufacturing of human protein pharmaceuticals and as stem cell therapies. But we would like to make them virus resistant, prion resistant. I actually have some close friends of mine who have inherited prion disease. We want to make them free of viruses, and I'll show you this in a moment, that these viruses that are built into your genome. And the list goes on. Radiation resistance might be of interest to space exploration, cancer resistant, and so on. Now, to do genome writing or editing, there are 9 nanomachines. These are small enzymes that can be combinations of DNA, RNA, and protein. And what they do is they scan through the genome, your 6 billion A's, G's, and T's in your genome. They will scan randomly, and they'll make a lot of mistakes, but once they find the right place, they'll make mistakes in scanning, but once they find the right place, they will make a cut, or they will sometimes swap and do a recombination, it's called. And 3 of them do that needle in a haystack search through 6 billion different places by using DNA interactions. So Watson and Crick, A base pairs with T and G with C. 2 of them use RNA with the same rules. And then 4 use protein DNA interactions to find that needle in a haystack where they're going to do the magic of editing or writing. The latest improvement, I wouldn't say necessarily breakthrough, which is CRISPR-Cas9, but the latest improvement was a 4-fold improvement over the previous one. Collectively, it's a revolution. There's no doubt about it. We don't need to go through the details of this data, but it was a 4-fold improvement in efficiency. We still have 2 major challenges in genome editing. One is off-target, and the other is on-target. In other words, we don't want this sophisticated nanomachine to be cutting off-target where it can get into trouble. In particular, if it cuts off target in a tumor suppressor gene, you could conceivably get cancer. Since the computer is involved in designing these things, and this is part of the intimate dance between reading and writing genomes, is you don't just myopically look at what you want to edit and say, I'm just going to edit that, I'm going to ignore the other 6 billion base pairs. You actually say, if I'm going to edit here, is the 20 ACs, Gs, and Ts I'm going to use to recognize that sequence, the computer is going to design that, is it matching anything else in the genome? Because if it is, that's a bad thing. And in fact, if it's off by 1, it's still a bad thing. If it's off by 2, it's still bad. Off by 3, you can tolerate that, although in best case, you test it to make sure that the off by 3 is adequate. We even have a version of this which is single nucleotide specific, SNP specific. That is to say, it can tell the difference of 1 base pair. But in general, you want it to be off by 3 or more. So that's one of the ways we deal with, 2 of the ways we deal with off-target issues. One is computational, the other is SNP-specific, so-called tuned guide RNAs. Now, on-target is even more vexing. You would think, oh, on-target, that's good. We're close. You know, that's what it's doing. But what happens is, instead of precise editing, where you say, I want this G to change to an A, it makes a mess. I call it genome vandalism rather than editing. For some purposes, that's what you want, but for many, that's not what you want. So we're working on ways to go beyond CRISPR, and 2 of them are in this slide here. And what they have in common is, rather than making a cut and then hoping that the correct sequence that you've also introduced makes it to that cut before the cell makes it. The cell is panicking as soon as you break its DNA. It's saying, I got to get that thing back together, and it'll bash it together and make a mess. But a mess is better than a double-strand break. Meanwhile, your donor DNA is kind of slotting along there randomly, and it doesn't get there in time. But the 2 that work well is they require that the donor and the acceptor be paired, they touching each other, 2 DNAs are touching each other before it does any editing. And that results in very precise editing, and I think that's what's on the immediate horizon. So if all of you have heard the hype about CRISPR, you can one-up it by saying there is these integrases and recombinases on the horizon. In fact, they're more than on the horizon. We have already used them for the largest genome engineering project ever, 2 of them actually. One of them is in yeast, and the other is in E. coli. Both of these are industrial microorganisms, as you probably know. They're very useful. E. coli has a particular problem with viruses, as do other industrial microorganisms in the dairy industry and in the mammalian cell culture. And so one of the goals of engineering the genome, and I don't mean just engineering a gene, but every gene in the genome, is so that we can make it resistant to all viruses. I think this is very profound philosophically, because we're not only we can make them resistant to viruses we haven't even studied, we haven't even seen before. Scientists don't need to have studied them, because viruses have one thing in common. They have many differences. They have one thing in common, which is they expect the host to provide the translation machinery that goes from DNA to proteins. And that is nearly universal, and it is certainly expected to have a certain properties, the genetic code. If we change the genetic code, we can make it so it doesn't impact the cell, because they're synonymous codons, these triplets of ACs, Gs, and Ts, triplets that are synonymous, code for the same amino acid. So we can make the cell accepting, but the virus wasn't in on the game as you change the code. So anyway, we've now done that, changed the code genome-wide in one organism, and we're in the process of changing it more radically and changing it in other organisms. This is one of the other organisms. The previous winner for the race for the fastest replicating organism, E. coli, which you've all heard of, is on the right there. And the new winner, which might be a little alarming to you, is Vibrio nitrogens, which we've now engineered to be almost as good as E. coli at everything that we consider the standard molecular biology toolkit. Plus, it's better than E. coli at replicating, as you can see from that little movie. And a lot of what we do is aimed at safety. Biocontainment and genetic and metabolic isolation was one of the things that comes out of that genome engineering, genetic code change. Now, when we start to apply this Genome Project right, whether it's editing or larger scale writing for the human genome, we have to think very deeply about testing as well. And this can go in two directions. If you think of, so now many of the things in our genomes, many of the DNA sequences in our genome are neither deleterious nor normal. They're just different. They vary a lot. Some of them don't matter. Some of them produce diversity that's wonderful. But let's take a case where you have a disease allele, which we'll call deleterious. And we can, with genetic writing, we can change the deleterious allele to the normal allele. And we can do this in an adult in the correct tissue, and that's typically called gene therapy. And we have a gene therapy test pipeline, and you'll see what I'm talking about in a moment. Almost exactly the same pipeline can go from normal cells to deleterious mutations in those genomes. And this is addressing a growing diagnostic need, which is that as you sequence everybody in this room that isn't sequenced and everybody in the world, you find a large number of things that look potentially pathogenic or alarming. Now, there are 3 million differences between you and me, and most of those are neutral. But you don't necessarily know right away, so you need a way of quickly determining which ones are serious and which ones are not. And I'll show you that in just a moment. What we need in order to do this, we need properly consented cells. We need a way of doing not only genome engineering I've been talking to you about, but epigenome engineering, meaning turning the way the cell is expressed on and off to make a particular cell type, because the mutation may affect blood cells, but not skin cells or vice versa. And finally, I really want to emphasize this. You need to have genome sequencing before you do it and after to make sure you got the right thing. Now, this is the epigenome engineering. You start out with a little pig pile. You saw how closely packed they were in little dots of cells. They loved each other as stem cells. These are stem cells from my body, by the way. No embryos were hurt in this. And no students were impacted either. Alex is a graduate student. And what's happened here is those stem cells that love being together flew apart and became neurons of a very specific type, bipolar neurons. And we can make neurons of many different types. And many other things that are present in the brain that are not neuron, which includes the cells, the blood vessels, cells around the blood vessels, the so-called glia, and the cells that are responsible for putting a myelin sheath around your axons to make super fast transmission of electrical signals. And we've learned how to do this. We made a library of factors that help us do this in a very systematic way. Now, I mentioned those cells came from me, but we have other cells and other data sets which are open, and I'm very indebted and delighted to be part of this sharing community globally in 5 different countries. And these are some of the volunteers in that project, which is the only open access project in the world where people share their genomic, environmental, and trait data, as well as the cells for projects like the ones we've been talking about so far. That project, the Personal Genome Project, has been recognized by the NIST and the FDA here. This town should appreciate exactly how important those 2 institutions are. And it's not every day that they collaborate with each other. And they did in this case to establish a genome in a bottle project to provide standards worldwide for the if anybody needs a genome, they want to be able to use the same genome that everybody else in the world is using. So there's a standard genome, standard cells, standard data. And so anybody that develops a new technology, a new diagnostic procedure or instrument can all get these standardized cells. And they found that our project was the only one in the world that the individuals have been properly consented for this kind of project. Now here's what I was getting at earlier about how you can run it one way where you go from deleterious mutations to normal, that's gene therapy, running it from normal mutations to deleterious. It allows you to get at variants of unknown significance. It allows you to determine whether a mutation you think looks dangerous in a diagnostic setting is or is not in a particular tissue. So in this particular case, this little boy had severe cardiomyopathy. These methods, I should mention, work with as little as an N of 1. You know, you often will hear that you need to have 10,000 patients, 10,000 controls in order to get good correlation. This goes beyond correlation to cause and effect. You can now get very convincing data, in a way more convincing than correlation, by showing that a particular base pair, not one of the 3 million differences between me and this young boy, but one base pair. And so what we did was we took my cells, which are ostensibly normal, although I assure you they're not, and changed one of the top candidate positions. So this 1G on the X chromosome and the TAS gene was deleted in this boy. And although there are many things that could have caused the various problems, that was one worth testing, and that's what is illustrated here, is that we made a 1 base deletion, removed that G, didn't affect the other 6 billion base pairs, and we know that because people in my lab think nothing of, if they change 1 base pair, they sequence the whole 6 billion to make sure that they change that 1 and nothing else. It tells you really how far we've come from the days when we used to agonize as to whether we could afford to sequence 20 base pairs. We now think nothing of sequencing 6 billion. And it also illustrates how that even though all the off-target, on-target things I was complaining about a little while ago, if you have a procedure by which you can get a clone, meaning that you can take a clump of cells, like I showed in that movie earlier, and disaggregate them into single cells, and each of those single cells produce its own little clump of cells, then each of those cells is more or less equal within the clones, and if you analyze it, you can determine the sequence of the rest. And that allows you to get nearly perfect editing, and that's what we have here, is we have a clone that is perfectly edited. That's very different than many of the other gene therapy scenarios where you will treat 1 billion cells, and each of them has a slightly different history and outcome. I think this is a very promising way of not only doing the diagnostic search, but also doing therapeutic. Anyway, we changed this 1 base pair, and then we used, that's the genetic engineering part, and then we did epigenetic engineering, where we turned those stem cells from my left arm into cardiac tissue. So this is now this beautiful repeating structure, sarcomere repeat, just like cardiac tissue. This thing beats spontaneously if it's the normal. But if you change 1 base pair, you now simultaneously change the lipid biochemistry, the mitochondrial function, the morphology, and the contractile nature, all in ways that are consistent with the medical phenotype of this young boy. So this consisted of pretty convincing proof that 1G on an X chromosome in the TAS gene was sufficient to cause this genetic disease. So that was something you can do with information from 1 child. A dirty little secret about the human genome is we have never sequenced the human genome. I'm sorry you heard it from me, but many of my colleagues have claimed that we have, or obfuscated the fact that there are parts of the human genome that are not yet sequenced. Many of the hardest parts are in the repeat regions. These are regions where you have the same stretch of sequence over and over, sometimes as short as 2 base pairs, sometimes as long as millions of base pairs over and over with slight variations. We hope to finally sequence the human genome. I won't declare victory until we've sequenced it many times over from many people. We haven't done it from 1 yet. But anyway, our lab is not only sequencing these hard parts of the genome, but we are engineering them as well, because I think that's how you get true understanding of such regions. And many of these are involved in cancer and aging, and so that these are not junk DNA. You know, my colleagues might have a sour grapes attitude, which is if they were so important, we would have sequenced them by now. And here's first example of us engineering repeat elements genome-wide for a very valuable reason. And this is, it turns out almost every mammal has endogenous retroviruses in the genome. This is not a hygiene thing. This is not something where these pigs, in this case, caught this from the environment. This is something that all pigs are born with these porcine endogenous retroviruses. That's not my acronym. And this is a phylogenetic tree showing how distant related some of these endogenous retroviruses are. We picked the most conserved part, meaning the part where the Gs, As, Ts, and Cs have changed the least over these millions of years that these endogenous retroviruses have been co-evolving with the major part of the pig genome. And we picked a highly conserved part in the polymerase gene. We didn't want to touch the envelope gene, which is thought to be involved in placental health during the development of the pig. And then we eradicated them. And you might be wondering, why are you, okay, I understand that you have this thing about repetitive elements and finishing genomes, but why are you doing this particular one? And the reason is that the humanization of pigs was a very promising technology 20 years ago, long before we were involved in this field. And it was shut down correctly by the FDA after about $2 billion investment because of these endogenous retroviruses, largely. These would leave the pig organs, in principle, and go into the immune-compromised recipient. You're often immune-compromised if you're a transplant recipient of any type. And then you could imagine that these pig viruses, which don't hurt pigs, will infect, they don't infect pig cells, but they will infect human cells, and then it will spread from human cell to human cell because they're not prepared to be resistant to them. So that was considered a showstopper by the FDA, and I think quite correctly. And so when we entered the field with our little CRISPR scissors, we thought, well, let's face that up front. While we're doing the humanization of the immune system and the carbohydrates and the complement and so forth, let's make sure that we're not going to have this showstopper again after 20 years. And what really surprised us is that it took 14 days. After 20 years and $2 billion, it took us 14 days and some cheap growth media to eliminate all 62 endogenous retroviruses from the pig genome. So these are the first pig cells in the world to not have any active endogenous retroviruses. And then we've gone on to do this again and have taken them through development. So, and we also have engineered the immune system, so hopefully it will be compatible enough so that now we hope to do transplants into humans within a year. And this is something that could affect millions of people worldwide. For almost every organ we currently transplant from human to human, including hearts, kidneys, liver, lungs, intestine, eyes, skin, heart valves, and so on. Now, one thing I like about this, aside from the obvious helping of millions of people, is the way that it makes us rethink preventative medicine I mentioned earlier. Because if I want to develop a radical new preventative medicine on you, the healthy people, that's hard because really I can only do harm to you in an immediate sense. I might promise you something down the road, but the immediate impact is potentially negative. So it's hard to do that. On the other hand, if you need an organ transplant, then it behooves me to give you the best possible organ I can. And so I can offline, without affecting any humans, I can develop pigs which are capable of producing organs that are slightly better than merely missing their PERVs and humanized immune system. And that means that we can make, in principle, we can attempt at least to make pathogen-resistant, cancer-resistant, and aging-resistant pig organs. And then we can get practiced at this, which I would consider preventative medicine. So we have a number of such projects going on, and there's evidence in the literature from a variety of groups in a variety of model organisms. Again, like CRISPR, it's the basic science that got us here, and then we technologists just pick up where the basic science left off. Is in fruit flies and worms and mice. You can find ways of getting longevity where you can get up to 2 to 10 times longer lifetimes depending on the organism. 2 to 10. Imagine living 2 to 10 times longer than the longest person in your family. And here's examples of some literature citations where, for example, blood from a younger mouse will rescue and reverse many different cell types and many different measurements of reversal. And this has been reproduced by many labs. There's also the same factors that we use for reprogramming my arm skin cells into embryo-like cells that I showed you earlier. You can do that, we do in the lab in the single cells, but you can also do that in a mouse, and that shows, and I'm quoting here, in vivo amelioration of age-associated hallmarks by partial reprogramming. So we're taking these observations from model organisms and we're moving them into gene therapies. Not into small molecule drugs, as might be the temptation of pharmaceutical companies, but gene therapies turn out to be much easier. We made 45 gene therapies in the course of a few months with just 2 postdoctoral fellows working on this. Based on some of the genes I've been talking about already, plus others in this aging gene database. And these have been tested in mice, and some of them have already moved on to being tested in dogs, where there is an interest in veterinary therapies, and it's much easier to get FDA approval for such things. I'll just show you one example. This is mitochondrial pathway. Here, mice, when they go from a young mouse, which is 6 months old, to a really, really old mouse, which is 22 months old, this particular chemical, this NAD involved in mitochondrial function, drops by about a factor of 2. And so we wanted to change the pathway by doing activation. I won't go through the details here, but the activation of a particular activator. So this transcription factor, TFAM, we used CRISPR not to cut it or to kill it, but to activate it. And we found empirically of 4 different places we checked, one of them improved at 48-fold. Remember, we're just trying to get a 2-fold effect. 48-fold in the protein and about 8-fold in the NAD, the small molecule that we wanted to improve by 2-fold. So this looks very promising, and this is just one of 45 different gene therapies that we're trying. There's a possible dark side to this, which is that some people aren't waiting for FDA approval. So these are 2 people that are well known. I'm not outing them or anything. They have been published by Tech Review, who independently of each other and independently of me, in fact, I discouraged them from doing this, and not necessarily even with deep scientific know-how in one case, just ordered on the internet adeno-associated virus with containing genes involved in aging. These 3 genes mentioned here, follistatin, telomerase, and growth hormone releasing hormone, and injected themselves with it. They fortunately did not die, and I think I take this very seriously. But it shows how far we've come and how cheap these things are becoming, and how small the barrier is to trying it out. I also show on the slide that you can do, your kids are out buying CRISPR kits for $150 and probably less. And these kits assume no existing equipment, even assume you don't have access to gloves. So there's gloves there. So do you know what your kids are doing right now, right? There is the inevitable discussion of germline therapy. I had been not advocating, but describing a potential slippery slope or possible that might be particularly attractive. It was easy. It's too easy. In fact, I caution against dismissing a new technology. Say, we don't need to worry about that because nobody wants to do it. Or we don't need to worry about that because it's not technically feasible, both of which were used for germline therapy. And I think that was, that doesn't allow us to address in advance. It puts us in a position where the technology advances very quickly, and we're not ready for it. So the scenario that I created, I think is more realistic and deserves attention and was eventually given attention in the National Academy of Science report that came out just this past February, is the idea that if you have parents, let's say 2 unaffected carriers, they have one good and one bad copy of every gene. Bad, I mean deleterious for a very serious genetic disease. And they, about a quarter of their kids will either die or be affected for life with this disease. The standard of care right now is either abortion or in vitro fertilization, where you might make 10 embryos and select one of them, which for right-to-lifers is the equivalent of death of the embryo. And these are not entirely acceptable alternatives. But what if you did engineering of the sperm or the eggs, the gametes? You're not putting any embryos at risk, in contrast to the way it's normally framed, and you are and you're not even doing anything in vitro at that point. The testes are modified in an outpatient clinic setting, and you have the sperm are produced, and the whole process of reproduction looks from that point on to be like normal reproduction. A second example where we can be too rash in saying this is not technically feasible, this is not going to happen, it doesn't accelerate, doesn't help us consider the risks if we dismiss. And here the dismissal is of so-called multigenic or complex traits. The textbook case is height. It's not exactly a terrible medical malady, but it is medically treated. There are about 6 different diseases which is treated by a single gene, growth hormone or somatotropin, which is, in spite of the fact that there are thousands of genes involved in this complex trait, and there are many hundreds of environmental components. Same thing's true for cognition. There are 3 genes that individually work one gene at a time that have very profound effects on cognitive tasks in mice, even though we know that in mice and humans there are hundreds of genes, just like height, that impact it in many environmental components. So to dismiss these and say, oh, it's so complicated we can't understand it and it doesn't affect the biology, I think is naive and too much dismissal. So I just want to end on the topic of gene drives. This is another example of something where we have to take it seriously. We have to talk about it broadly. We want to include the entire community. I think it's very important that we have more business and political leaders who are very savvy about science. We need more scientists that are very savvy about business and politics. We need to have a discussion of everybody, not just those people. And what it is in this case, the stakes are very high on both sides. We want to release into the environment genetically modified organisms. Now you could say, oh, that's a non-starter. People don't even let genetically modified organisms onto the farm, which is kind of not really an environmental release. Putting them into wild animals. I think the difference is that even anti-GMO forces are quite accepting of medical dire need. So ask whether anyone is opposed to genetically modified insulin produced, human insulin produced in bacteria, or erythropoietin, or a variety of human protein pharmaceuticals that are not made in the original organism. These are saving millions of lives. They are very different from genetically modified crops, where the benefit to society is less obvious. To the individual in the grocery store, it is not obvious. Here, again, like genetically modified insulin produced in bacteria, these are cases where you have an insect or other vector, like a white-footed mouse for Lyme disease, the Anopheles gambiae for malaria, Aedes aegypti for dengue and Zika and so forth. We would like to make them resistant to these pathogens. Some of these pathogens are human-specific, so if we can get rid of them even momentarily, as happened with smallpox, then we need never worry about vaccines, drugs, or even gene drives in the future. Many of the things that we're targeting gene drives for, vaccines and drugs, have already been tried unsuccessfully, sometimes for many decades. How this works, and very importantly, when we proposed this in 2014 in this paper, we didn't just do the experiment and then plop it on society the way it was done with gain-of-function mutations that cause more serious influenza virus. We said, let's have a discussion. This is the way we can do it. And more importantly, can we make them reversible? We don't want to talk about a technology until we have some way of making it reversible. There are ways we can have a consensus building, not just among the human community, but among the animals, so that if you're doing an island, for example, you can drive it to extinction, while on the mainland it will not happen. And you can make it local in various ways. And so the standard drive, this is CRISPR here indicated by scissors, will cut a DNA, and then this drive, as this blue bar, will copy over and fix that. And then that repair, now all the offspring have inherited, rather than normal, which is 50%. So instead of like staying at some low level, whatever level you introduced it at, it exponentially grows and then plateau, completely fixes, takes over. To reverse it, one way to do that is just to put in another drive that replaces the first one, and it will follow the same time series. We have another more complicated way where we can keep it constrained in space and time, and this is work from Charleston Noble and others in our consortium. We have 4 professor groups that are working together here, and, where one CRISPR gene drive runs another in a different chromosome, and then a third one, and none of these things will spread by itself the way a normal gene drive will, but we call it a daisy chain, where one is at low levels, it causes the second one to be at higher levels, the third one to be at higher, and eventually they all decay, but at different spreads. So you can have something that's local in space and time. So that's an example of how you can discuss these things in advance. We've tested many of these things safely in the laboratory before we even get close to making a decision as to whether these things should be released. But the benefits are quite high. 600,000 people a year dying of malaria. I'm sure some of you know somebody who doesn't like Lyme disease. And the list goes on and on. So I just want to open it up for, I know we're going to have lively discussion here. These are some of the people that have helped me do the genomically engineered organs and work on various aspects of schizophrenia, bipolar, and so forth that I didn't talk about. Thank you very much. I think there are microphones here and there if you have questions. Or you can shout it out and I'll repeat it. Yeah, I'll repeat it. Right, so just rephrasing the question a little bit, both so everybody can hear it and so I can rephrase it. How general is how universal is the genetic code, and if it's been fine-tuned over so many years, who are we to try to improve on it? fact is, it's more than just every vertebrate sharing a genetic code. We can move human genes into bacteria. That's about as far as you can go, and they work pretty well. We share that genetic code of the. There's some small exceptions, but for a class of viruses, you know what code it's expecting. If you take that code essentially offline, when the viruses aren't looking. So normally, what would happen in evolution is the code could change, and the viruses would keep up. But if you take it offline, you change it in a way that doesn't hurt the organism, the host, and then let the virus come in. It can be so radical a change, so many different parts of the virus are broken that it can't even mutate around it. But back to your question is, will that work for the host? And the answer is we've changed one organism radically enough so that we've made the first organism in the world that has only 63 of the 64 triplet codons. It doesn't have the last one. And it seems to behave fine. When we originally made it, it had a little bit slower growth rate, but it turned out it was not because of the code, but because of mistakes that we made along the way we cleaned up. Since then, we've also put that codon back, but as a new amino acid. So now it has 64 again, but it's a new amino acid. That also is healthy, and these are multivirus resistant. So my gut feeling is that we can pretty radically change that genetic code to make it not only healthy, but healthier in a competitive environment, which includes infectious agents like viruses. And I think that this strategy should work for every organism that currently has a virus problem. And it includes a lot of industrial, agricultural microorganisms, plants, animals, and even humans, or at least human cells used for manufacturing and therapeutics. Yeah.
Well, first I was interested in the woolly mammoth project.
Yeah, sorry I deprived you so far, but I can make up for that. Yeah.
Oh, okay. I was wondering whether they were house pets or what you intended.
Okay.
But anyway, first of all, it would seem to me the most efficient place to be doing this is before fertilization, human or otherwise, because you have more leverage. Okay. So you didn't get into the mental aspects a lot, but my question is, what in your imagination could the human being, in terms of appearance and experience and mental lifetime kind of experience, what could, in other words, what could come of this that's pretty much realistic? What could we expect to see in a future lifetime?
So I think you're asking sort of the enhancement question, but from a particular angle, which is what's a realistic set? For the moment, not being judgmental as to whether it's desirable or not, but where could it go realistically? Although it is we'll accept that it's speculative. And then I will answer the mammoth question as well. First, the mammoth, they're not intended to be pets necessarily. They are, in my opinion, they're like an Asian elephant. They're very closely related. They probably would interbreed. Asian elephant's an endangered species. One way, and it's mostly endangered because it is in conflict with humans. It's in high human population density. If you could give it the boost of its close relative in ability to proliferate and thrive in cold environments, it now has access to one of the largest unhuman populated ecosystems in the world. It's just this vast tundra in Canada, Alaska, and Russia. And it would benefit the Asian elephant and potentially and the environment itself. So that's where I see it, not necessarily in zoos or homes, although I have had many requests for pygmy mammoths, which is almost an oxymoron, right? Yeah. But back to human enhancement, which is a sort of a related topic. We're enhancing Asian elephants so they can live in the cold. Most of the things that you would want to enhance humans, we've already enhanced with physics and chemistry. There's no particular reason to make me run faster because I can hop in a 747 or a, Volvo or something, and I can go faster than any reasonable runner. What is left that we can't fix with rockets and electromagnetic spectrum and so forth is resistance to pathogens, senescence, and things having to do neurocognitive. And those are things, and even those, there's a little bit we can do with physics and chemistry and biochemistry already. But imagine being resistant to all pathogens. I mentioned the aging resistance. These will be framed, they won't necessarily be framed as enhancements. They will be any more than a smallpox vaccine or polio vaccine is not framed as an enhancement. It's something that's dealing with a very serious disease. Aging is 90% of us in developed nations will die of a disease that typically doesn't kill 20-year-olds. It's a disease of aging. And so this will be framed in terms of disease, but it most certainly will make us able to survive in a new way. Now, the behavioral and cognitive is the biggest thing. We can augment that with education, with computers and cell phones and so forth, Google, Wikipedia, but our brain could itself contain a lot of this information. It could contain new ways of dealing with it. And I don't know where that goes, but that's one possibility for something that would be. There are a few others that might make us particularly suited for space exploration, like dealing with microgravity and radiation. Again, those might not be considered enhancements because you're going to be very sick if you're in that environment. So a lot of it has to do with what is the illness of the environment.
Would we expect to live longer? I mean, we'd have a more crowded planet.
Right. So if we did, let's say, get aging reversal to work, and I think that's a more reasonable FDA approval process than a longevity drug, because if you go to the FDA and say, I have a drug that will extend life by 30 years, they say, great, come back in 30 years with your data. But if I say, here's a 2-month trial that shows increased, grip strength and reaction time and so forth, then they say fine, or resistance to heart disease. So if that resulted, which could be initially framed in terms of dealing with diseases of aging in an acute sense, but it could result in longevity, not immortality, but just much longer lives, we could have a population problem. On the other hand, look at the cities of the world, all over the world. Even countries that have high population growth have cities that have negative population growth. Japan, as a whole country, has a problem with population growth, and so do in France and a bunch of others. Most of the ones that would have an even bigger problem if it were not for immigration. So the fecundity drops from about 7.5 outside the cities to about 1.2 inside the cities, where 2.1 is the replacement rate. So that's one partial solution. The other one, presumably, would be colonization of other planets, which, if we solve poverty and aging, we might have some excess wealth to spend on what doesn't have to be that expensive, an enterprise. Charles can tell us about that.
Thank you.
In the middle there. Before you get to your other two questions, let me just repeat this one and answer it, okay? And we might let other people ask some questions too. So the question is, do we have, how does this cause and effect, how is that better than correlation? And under what circumstances, and how do we ensure that that's actually working? So let me start with the circumstances where it won't be better. I mean, there are some things that are very hard to test in organoid systems such as the ones I've been describing. The organoids are getting better and better. They're getting more and more like fully functional systems, but there's some diseases that really require a fully functional integrated human being, and vary in very subtle ways depending on what you ate that morning and so forth. There's this whole neuroendocrine loop that's very hard to simulate so far. But I've been amazed at how quickly not only these omics methods are going, genomics methods are going, but also how quickly the organoid methods are going. So I don't know, but there will be many cases where the disease has a molecular, a cellular, and an organ level phenotype, where you can see what its effect is. And for those, you will be able to have a possibly very high throughput pipeline, where as soon as the data comes in that day from all the patients, it goes into maybe several facilities around the world, and they go, and out comes the consequences. That's one scenario, but we'll see.
Yeah,
person at the microphone here, and then you.
Yes, you had mentioned that you had advised Liz Parrish against her experimentation, but given that some time has elapsed and she seems quite pleased with her results, have you changed your opinion about that?
Well, my opinion wasn't based anyway on her personal plan. It was based on, in this case, we do need something a little more than an N of 1, because we're not doing a cause and effect analysis on a person. I mean, it could have been, if it had been a very dramatic, effect, like she turned into Spider-Woman or something, then N of 1 would have been adequate. But for most realistic, even amazing blockbuster drugs, you need to have a pretty big control cohort. You need randomized clinical trials, double-blind placebo control, ideally, because there is a potential placebo effect on both of them, on feeling stronger, feeling healthier, and so forth. So I think that would be my critique, and it stays in place. We need, and now the other critique would have been that the trials were not done with animal trials first, but they were actually both done in animal trials, just not exactly the same drug formulation. But it's actually hard for gene therapies to have exactly the same formulation. It's not like a small molecule drug. In particular, gene editing, which is not what they were doing. Gene editing is very dependent upon the background genome, not only what you're targeting, but what's off target. In any case, I mean, I think that it's a bold move. There will be required many well-funded studies to follow up.
But isn't someone like her moving the field forward by taking that kind of risk on herself?
I wouldn't say she could put the field backwards by having a bad outcome. She might be moving it forward. She certainly, she might be. I think the field is moving forward very quickly, even before Brian and Liz started doing this. I don't want to in any way undermine what they're doing. I think it's terrific that they are engaged. I just think that they should raise enough money so they can do at least one control at a minimum. I think the field is going to go forward very quickly. I do, unless something bad happens. And so that was me worrying about them and the field, as happened with gene therapy. So one person died in Pennsylvania and 2 people died in Europe in around the year 2000, and that set the field of gene therapy back by 10 years. And you might say, well, 3 people die a lot in clinical trials, but they don't die a lot when a brand new technology is coming along. Anyway.
Thank you.
Right. So the question of gene therapy cures. So there are quite a few. There's 2,400 gene therapies that have been approved for clinical trials. A few of them are getting either have been approved, fully approved, made it through phase 3 clinical trials, or getting close to that. Some of them are curing people in phase 1, which is pretty amazing. Some of them cured so many kids of blindness in phase 1, there's almost nobody left because it's a very rare genetic disease. I think if we're cautious, gene therapy may turn out to be one of the most easily programmed, transition from basic facts to therapies, and very safe. But it's still not. I mean, it hasn't been proven in enough cases yet. So I think that we need to proceed cautiously. One last question back here. I'm sorry, there's a few hands, but yeah. So there are 2 questions here about eliminating viruses. And let's start with the easy technical one, which is if we made a cell that was resistant to viruses at the genetic code level, would it also be resistant to viruses where you're trying to deliver a payload? I'm not even talking about gene therapy, just in a very generic sense. And this could be any organism, bacteria, animals, whatever. And the answer is that typically when a virus is delivering a payload, that payload can be engineered to be whatever you want. It can be engineered to have the genetic code of the new cell, the new genetic code. Also, the virus capsid itself is already made before you deliver it. So it can be made with whatever code it wants, incompatible code, but the protein still works. So the code is only about a nucleic acid going to a protein in the recipient cell. And so you could have like a capsid made with the old code delivering a messenger RNA with a new code. So that all works. That's the technical question. Your slightly more philosophical or question that maybe we don't have enough data for is how valuable might viruses be? We can start with how viable microorganisms might be, and we kind of went from a world where everybody felt all microbes were bad. Well, first didn't even know about microbes. Then all microbes are bad pathogens, and then some of them might be good. To they're so good you can't live without them. That's kind of the history of this science, and I would say that went too far, because there are examples. Almost every animal we've tried, I mean, I'm putting aside maybe termites and cows, every major, goats, chickens, rodents, and even humans, you can eliminate all their microbiome as far as we know, and they'll survive. So it's not absolutely essential for life. I wouldn't say necessarily we've settled whether they thrive or not. I mean, they're probably fine. Viruses, they might be in there and we're not noticing them. I mean, that's a reasonable way out. But I think that if that were the case, then you could probably, if you find that virus, it's not like a vast number necessarily. It could be one virus, one bacterium that's really helps you to thrive rather than just survive. You could probably simulate that with either a vaccine or that pure virus. But that's speculation. The point is there is definitely some ignorance there. It's not vast ignorance, but there is ignorance. So.
Dr. Church, on behalf of, can you hear me? On behalf of the Council of Scientific Society Presidents, the Cavalier Foundation, and the Carnegie Institute for Science, we thank you for your talk. We thank the audience. We thank the audience for their attention, and I will tell you that normally we would have turned the lights up, but the same gremlin that had affected the microphone has apparently infected the lights, and we're afraid if we turn them up, we'll lose them entirely. So be very careful as you leave. All right? So thank you for coming. Good night.